Okay, great. Hello everyone. Welcome to PAPERI webinar series. My name is Mariam Youssef, and I'll be today's moderator. Thank you for joining us for our session on AI-driven network medicine applied to emergent infectious disease. I'm delighted to introduce our esteemed speaker, Dr. Hilder Nakaya. Dr. Nakaya is a senior researcher at Hospital Israelita Albert Einstein in Sao Paulo and an associate professor at the University of Sao Paulo, Brazil. He holds a PhD in molecular biology and has extensive training in bioinformatics. An expert in systems immunology, he works at the intersection of system wide measurements, networks, and predictive modeling, particularly as they relate to vaccines and infectious disease. His lab concentrates on investigating the foundations of infectious diseases through computational systems biology. Additionally, Dr. Nakaya serves as an adjunct professor at Emory University School of Medicine in the Department of Pathology. His seminar will explore the recent advances in AI-driven network medicine and its application in addressing the challenges posed by emergent infectious diseases, providing new strategies for combating these global health threats. In today's talk, Dr. Nakaya will discuss emergent infectious diseases like Zika, Chikungunya, and Dengue that are often driven by complex interactions within molecular networks. Network medicine investigates these intricate intracellular and intercellular connections to uncover the underlying mechanisms of diseases. By integrating AI with network medicine, we can better understand the complex system, leading to innovative approaches in precision medicine. Please join me in welcoming Dr. Hilder Nakaya. And please note that we're going to have a live Q&A session at the end of the webinar. You can write down your question in the chat box to submit your questions. Dr. Nakaya, please, the floor is yours.
Thank you so much, Mariam. Hi, everyone. I was telling Mariam that I wish I was there physically because Egypt is one of my bucket list place to go, but maybe next year. So today I'm going to talk about AI-driven network medicine. But first, I would like to explain what is network medicine, why network medicine is relevant for health bioinformatics. Well, it is because of this. If you get a group of people that is infected with the same pathogen or that receive the same vaccine or treated with the same drug, they are going to have completely different outcome. Some are going to be fine, some are going to be sick, and some are going to even die. And that happens because every single one of us has a unique immune system. We have different age, we have different stress, we have different genetic backgrounds, and everything influences the outcome. Okay, so today we are able to measure the activity of millions of biological components, all the transcripts, all the proteins, all the metabolites, and we can create networks like this. And what we do in the lab is actually try to find what is the smallest piece of this network that is able to predict the outcome. So that's in a very simple way is network medicine. Well, and this has to do with this misconception here. For a long time, people tend to focus on the gene, protein, or metabolite that is changing the most. Oh, which biological molecule is changing tenfold increase, and that's going to be the most important feature for the outcome. But this component here that changed tenfold is less biologically important than this one that's only changed fivefold, but is connecting three different modules here. Same thing, even this group of genes or proteins that are changing just a little bit, 20, 30%, is actually more biologically relevant than that gene that was changing tenfold. So network medicine takes into account not only the change in the biological levels, but also the context, the network that is behind. And throughout this talk, I want to focus in how can we do low budget, high quality science, because this is important for the global South, including South America and in Africa, because most of the studies that I'm going to show here was very, very cheap to perform. Okay, so most of the examples are going to be very low budget. And that's not something new because scientists do science with low budgets for thousands of years, right? So when you probably the philosophers, the Egyptian, many thousands of years ago, they were looking at the sky or looking at nature, observing, and then making hypotheses and testing the hypothesis. What is expensive here today is the amount of money that is invested on you to become a scientist. So if you are trying to look things that are very far away, that's going to increase the costs of science. So if you want to take a picture of the black hole, that's going to cost $55 million. And of course, if you want to look things that are very, very close or very, very microscopic, then it's also going to cost a lot of money, $23.5 million per year just on the electricity costs. And if you want to look at many things in parallel, that is also going to increase the cost. So if you want to do spatial transcriptomics of the whole organism, that can cost more than $10,000 per sample. So that means that generate data is very expensive. Storing the data is very expensive. And training people is very expensive. The part that I want to show today is this part here. The low budget part is once the data is already collected and stored, and once we already have people that know how to analyze the data, then we can do like very good quality science without spending more money on this. And I hope I can convince you at the end of the talk that this is possible. Over the years, we have been published several studies applying this approach of network medicine, systems biology on different disease, infectious disease with chikungunya, Zika, yellow fever, influenza, Ebola, but also psychiatric and neurological disorder, inflammatory disorder, and even cancer. Zika was something that we published a few years ago with a lot of researchers here, and we look at the brain of babies that die right after birth and that has congenital Zika syndrome, including microcephaly. So we performed transcriptomics, proteomics, and genomics of these brains, and then we could integrate all of this and create what we call the molecular networks that probably is associated with congenital Zika syndrome. Then we validate and look at the histology and see that, in fact, this is what was happening in the brain of these babies. And when I was showing this network in a conference, one of the researchers in Brazil, Mauro Teixeira, he was surprised that I was mentioning glutamatergic synapse as one of the pathways associated with congenital Zika syndrome. And the reason for him to be surprised is that he had a hypothesis that glutamates may be involved in the congenital Zika syndrome. Glutamates, in addition of being amino acid, is also a neurotransmitter. And when you have excess amounts of glutamates, this is neurotoxic. So his hypothesis that Zika was altering glutamate pathway, and that's leading to neurotoxicity. So how can you prove that this is true? How can you test this hypothesis? Well, you can use a drug that is an antagonist of the receptor for glutamate, memantine. And Mauro picked this one because this drug was used for treat Alzheimer. It's also safe to be given to pregnant women. And then he wants to see if it prevents neurotoxicity. So the first experiment he did was to get primary neurons infected with Zika, and then he showed that neurons die and the virus replicates. But when you do the same experiment, but now using memantine, now the neurons do not die and the virus replicates. So he kind of like showed that maybe you can reposition a drug that is safe to be given to women to prevent congenital Zika syndrome if they were infected during pregnancy. Of course, there is a long way until this is licensed or even tested in human, but he has other publications supporting this hypothesis. For yellow fever, this is a paper that we just got accepted, where we collected blood from people during the acute infection of Zika, of yellow fever, and some of them unfortunately died, and some of them survived. And then we did blood transcriptomics of during the acute phase and also the convalescent phase. And then we compare what is happening. Can you predict who's going to die of yellow fever if you get the blood during the acute infection, right after the person was infected? And then we look at several laboratory parameters, and then we saw what was the difference between who survived and who died. And we also performed transcriptomic analysis showing what are the genes that were involved in this process. Uh, we saw that antivirus and also plasmablast genes were involved. But what, and we compare with vaccination profile because the yellow fever vaccine is a live attenuated vaccine, and people thought that this was a mild infection. But we saw that actually there was several things that were very different during the wild type infection and the vaccination with live attenuated virus. Well, what I want to show here is that one thing that we did that was not possible a few years ago, which was using ChatGPT. So we had a list of 300 genes, and we knew that immature neutrophils were also involved with the people that die of yellow fever. But how can we show which genes were probably related to these neutrophils here? Well, we could do the one way, which was going to be copy and paste the gene in PubMed and then put in neutrophils and then put it in infection and try to see all the papers that were there and repeat this process hundreds of times. Or we can use ChatGPT to say, okay, take the list of differentially expressed genes and tell me which ones are related to immature neutrophils. And then from that smaller list, then we can do a proper human curation, read papers, and then see if the association makes sense. Chikungunya, we try to predict who's going to have chronic arthralgia after infection. And we use a machine learning algorithm called random forest. And I want to explain you how random forest works just to show you that it's very easy to understand machine learning. Well, random forest works like this. Imagine that you have 18 patients where you have 9 people with non-chronic and 9 people with chronic arthralgia. We have actually dozens of people in the paper, but I'm only showing 18 here. And then we have the transcriptome for each one of these people. And then we have 25,000 rows for each one of these people. How random forest works? You take the expression, for example, of gene A, and then you see where the dots are in this expression of gene A. And then you can decide for a cutoff that separates most of the blue dots from the red dots. And then you put information here. And then you do the same with these people here, but for gene F. And then you put the information here. And then you keep doing this until you create a decision tree. But the trick is that you don't create a decision tree from the entire data set. What you do is actually you select a subset of random people and a subset of random genes. And then with this random data set, you create a decision tree. And then you repeat this process 1,000 times, and then you have 1,000 trees. And what is the name of a place with 1,000 trees? Force. So random force is this. And then you can count how many times the gene appeared in these decision trees, because that's going to give you the importance of the gene in predicting an outcome. And this algorithm is very easy to run in any different context. But of course, it doesn't work for all the types of data. But this one works really well. And in the paper, we show that CD1C was the gene that was the most important genes in predicting who's going to have chronic arthralgia. And this gene is actually a receptor that binds to exogenous lipids in the dendritic cells that presents to CD8 T cells. So that raised a hypothesis that Allison tried to validate. So Allison reanalyzed the data, now looking at the T cell repertoire, and then he saw that the diverse of T cells decrease in the chronic people compared to non-chronic arthralgia, and the levels of T cells do not change. And then he compared this diverse with gene expression, and then he saw that there was a signature related to arthritis. That was really interesting because we could validate in 2 different independent studies. And then you may think, oh, but this would happen for any infectious disease. Well, when we do the same thing for dengue, only appears for the chikungunya data sets. And that was published very, very recently, showing how we think that chikungunya is driving the long-term arthralgia. With COVID, we also study different angles of the disease. We have more than 33 papers on COVID, and I'm going to show you some published and some unpublished papers. This one was a study that I like very much because it started with something that has nothing to do with COVID. It was something called Barrett's esophagus. And Barrett's esophagus is a condition that happens when you have reflux. And this reflux over the years change the morphology of the esophagus. So this is something that appears in 5% of the US population, 5% of the Brazilian population. So Leandro, in my lab, he was analyzing 8 transcriptomic studies of Barrett's esophagus, and then he found that when he looked at the 8 studies and then asked for the genes that were consistently up or down regulated in at least 7 out of these 8 studies, he saw that ACE2 was upregulated in Barrett's esophagus. And I don't know if you remember, ACE2 is the main receptor that the virus SARS-CoV-2 used to enter the cells. And that may give us an idea that maybe Barrett's esophagus was related to severe COVID, and nobody was looking because that has to do with the gastrointestinal traits. When we look at the pathways, we saw that regulation of intracellular pH was affected in this list of genes. And remember that I said that this has to do with the acid from your stomach going to your esophagus. This makes sense, right? But nobody, 0 people were investigating how the acid pH could regulate the expression of ACE2. So that's what we did. So we validate this using in vitro, showing that if you put human cells in a pH 7.4, this is the level of ACE2. If you put them in a acid condition, the increase of the gene, there was an increase of the gene expression. Okay, but what this has to do with the virus? If you infect the cells, you're going to see that cells that are under acid condition, they have higher viral replication than the ones that are in normal pH. Okay, what is the clinical implication for this? If you want to test this in vivo, you would need to recruit people that had Barrett's esophagus, gastritis, and that would take a long time. What we did was to use the fact that people that have Barrett's esophagus or gastritis or reflux, they take proton pump inhibitors, omeprazole, pantoprazole. And what we did was to go to the cohorts in Brazil that were already collected and then read the people that were treating or not for these conditions, that were taking or not proton pump inhibitors. And then we separate these people. And then when we look at one cohort in the Northeast, we see that people that do not take the drug survive more than people that take the drug. And even when you adjust for diabetes and hypertension, the risk of dying of severe COVID increase if you are taking this drug. And that was validated in a different independent cohort, showing again that the risk increases. And this was later validated in different parts of the world with publications. But that was very nice because we showed that this study started with analyzing public available data, completely 0 cost. And then that led to a hypothesis that we can validate in vitro. And then we went to in vivo and test this in patients. This year also came up with this paper here that was very nice, that shows that how SARS-CoV-2, the virus, could change the splicing machinery of the cells, in the antiviral genes, which are the type 1 interferon genes, were actually impaired. So what happens that interferon stimulated genes works to suppress the viral replication, but what the SARS-CoV-2 infection does is to do not change the expression of these genes, but it changed how the genes is actually spliced. So the cells produce a lot of mRNAs that actually do not produce proteins that are functional because they have unproductive isoforms of the genes. And then we show that several parts of this was actually impaired because of SARS-CoV-2 infection. This paper here, which is unpublished, that was led by Jonathan in my lab. And what Jonathan and the group did was to look at transcriptomic studies of severe COVID. Guys, there are over 50 transcriptomic studies available already. But the thing is that if you take two studies at random and then look at the genes that were differentially expressed, and then you compare the list of genes differentially expressed reported by the authors, you're going to see that there is a very little overlap, meaning that there is no consistency between these two studies. What we tried to do was to collect all these data sets, reanalyze them, and then to look at signatures that were consistently changing in these different tissues. So we look at not only one blood studies, but actually 8 blood studies of severe COVID. And then what Jonathan and the group found was 206 genes consistently changing in the blood, and apply the same thing for liver, kidney, hearts, brain, etc. And then we have a map, an atlas of severe COVID-19. Then we can integrate everything. So every dot is a different gene connected by the organ that is affected in severe COVID. And then we try to show that this resource is actually important for coming up with hypotheses and insights that are novel. Then we pick this gene here, RAB8B. And RAB8B is interesting because the RAB family has to do with the endocytosis of the virus and exocytosis of the virus. And nobody showed the role of RAB8B in the virus infection with SARS-CoV-2. So we pick this gene because it's novel, and then we validate showing that if you have cells with RAB8B, this is the viral replication. But if you remove RAB8B from the cells, the viral replication decreasing, meaning that this gene was important for viral replication. Then we also create a dashboard where you can type the name of the gene that you want, and then it's going to tell you which organ the gene is up or down regulated. So if you change the gene, you're going to see. So you don't have to redo 56 analysis to check if the gene of interest is changing in severe COVID. Single cell system biology. This is a paper that is also unpublished and started with Yuvka and now is led by Jonathan as well, where we have single cell RNA-seq of skin of people infected with cutaneous leishmaniasis. And then we have the cell types. But what I want to show you here, guys, is this. In order to create this figure that is part of the paper where we show what is happening in gamma delta T cells, we have to use, we have to look at 20,000 genes for each cell type. And then we can use bioinformatics to reduce the amount of genes by finding the differentially expressed genes on each cell type. And then we can also use ChatGPT to decrease this even more. But to create this figure, there is no other way if not looking at the genes, the papers, reading, studying, talking to experts, and then creating this figure. So that was a really nice exercise that shows that there's no program that you press a button and then magically this figure appears. So bioinformatics only serves for you to decrease the amount of things that you have to study, but at the end you have to study. And we also have been developing many computational tools over the years, and we are in page 2 already. I'm not going to talk about most of these tools, but just a few of them. This tree here has to do with machine learning. RANSAC is a tool that is you can check here that can perform correlation analysis even when you have outliers. So if you use all the dots, this is going to be the correlation, no correlation. But if you use the tool, you're going to see that in these data sets, there is a correlation because these guys here are outliers. BioFits works as a funnel where you can use as inputs any kind of data, cytokines, proteomics, microRNAs, and then the tool is going to prioritize which features are the most important in predicting something. And that is because we use feature selection machine learning algorithms. Feature Combinator is another tool that Juan and Andre is finishing, which combines the features and then tries to find models with good accuracy when you have combinations of features. Metatranscriptomics started with 2 guys in my lab, Andre, which was working with microbiome, precision microbiome, where he's looking, for example, what is happening in the microbiome of people with intestinal bowel disease and also other disease. And then here's to show that many different data sets. But what he's going to do now is making a graph neural network analysis where you can get the people and the microbes that are inside the people that has or not IBD. And then if you have an unknown person that you want to know if the person has or not IBD, we can use graph neural networks to predict this. And also the feature combinator parts. Icaro was helping with the integration of microbes and gene expression. So what we did was to create a pipeline where we take the reads from the RNA-seq, bulk RNA-seq, and then we can map in the human genome. And then what people do is usually throw away the unmapped reads in the garbage. What we want to do is actually to compare with microbes. This way, we can find that we are running already almost 60,000 human samples. This is actually the pipeline that's showing that it's not that simple. And Icaro has been applying this, for example, to study sepsis, where he looked at what is the composition of the microbes in the sepsis by looking at the bulk RNA-seq data of these patients. And then he found what is the contribution of having the bacteria versus not having with sepsis, and also between females and males with sepsis. Can we apply this approach to single-cell RNA-seq data? Yes. If you have single-cell RNA-seq, then you assume that some cells are going to be infected with microbes intracellularly. Then if you do the drop-seq, you're going to see that some of the RNAs from these microbes are going to be in this drop. And in theory, you could measure this. And this is what we did for IBDs. We took single-cell RNA-seq data from IBD patients, and then we look at the potential presence of microbes, and then they found that some of these cells were infected with some of these bacteria here. And it's, of course, it's a very low number because single-cell RNA-seq is does not produce many reads per cells. But it's very cool that when you look at infected B cells versus non-infected B cells, you do see several genes that are interesting and has to do with infection. This paper here is also unpublished and was led by these people here. Mayara is also from our group, and is working on cervical cancer. Cervical cancer is a cancer that has a slow progression and is associated with HPV. What they did was to look at, for example, bulk RNA-seq data of women with cervical cancer. And these are the different stages of cancer. And the authors also measure genotype, the HPVs that were associated with cancer or not. And then once we run our pipeline, we could see which bacteria and virus these women had. Each row here is a different women, and the reads from bacteria are in green, and reads from HPV are in red. And some of these women, the authors could not find any HPV, but we did find some reads matching HPV. But the most important part is when we look at the bacteria. When we look at the bacteria, we see that some women were infected with Treponema pallidum. And Treponema pallidum means that these women were with syphilis, and the authors did not know about that. And that definitely impacts the downstream analysis. So we found that HPV was associated, the levels of HPV was also associated with this Cutibacterium acne, which is a very common bacteria in the skin, but it's also a bacteria that is related to establishment and maintenance of skin homeostasis. So that led to a hypothesis that maybe this bacteria has to do with the slow progression of cancer, of cervical cancer, with HPV. Well, they validate this using another data set showing that, again, we could see women that were infected with syphilis, that had syphilis, and also Cutibacterium acne. We also look at proteomics, and then we do metaproteomics, where we found, again, Treponema pallidum and Cutibacterium acne. We validate this into another two independent bulk RNA-seq data showing again that this is true. But we compare the levels of HPV with the levels of 20,000 human genes to see what are the genes that were associated with having more HPV. And then we found, for example, that there were 700 genes that were possibly correlated with HPV levels. When we see the pathways that were enriched for the 700 genes, we see pathways related to DNA replication, mitosis, cell proliferation, meaning cancer. But we also saw this gene here, which is Fanconi anemia family genes, and these genes are very interesting because they had to do with DNA repair, cytogenetic instability. So these genes may be driving cancer. Someone already showed that HPV can induce the expression of Fanconi anemia genes, and Fanconi anemia patients have more risk of developing cancer. But this study here was done in vitro, and this study here was done in Fanconi anemia patients. And what they did was in vivo in women that did not have Fanconi anemia. So that was really nice. As a conclusion, Hippocrates, which is the father of medicine, already used to say that it is more important to know what sort of person has a disease than to know what sort of disease a person has. Meaning that even that time, it was more important to know the person, not the disease, because even if you have the same disease, you may have completely different outcomes. And we published a paper last year in PLOS Computational Biology that shows how can you use large language models such as ChatGPT for bioinformatics. And what I say to everyone now is that anyone can perform bioinformatics. And very soon, like 1-2 years, people are going to be able to talk to the computer to do the analysis. You're going to be able to say to the computer, I want you to split the data sets in these groups, and then to compare these two groups using the proper statistical tests, and make a graph with the x-axis with this information and the y-axis with this other information, and give me the results. And the ChatGPT or Claude or any other large language model is going to create the agents, perform the code, do the analysis for you, and then give you the results. And that means that bioinformatics, if you focus only in the bioinformatics part, you're going to be replaced. And there is a train coming in a very fast speed, and we cannot ignore that this train is going to arrive. But I also need to emphasize that having plenty of public available data sets out there and having bioinformatics tools and large language models available for you, that does not translate into low budget, high quality science. It's not simple as that. Okay, because we have the data, because we have the tools, let's make a nice science. And the reason for this is because of this book called The Hitchhike Guides for the Galaxy. In this book, a group of aliens, planets of aliens, they were tired of thinking about this philosophical question. What is the meaning of life, the universe, and everything? So in order to answer this question, they create a computer with the size of a city. And the name of the computer was Deep Thoughts. And then they turned on the computer and said, okay, Deep Thoughts, can you please answer what is the meaning of life, the universe, and everything? And then the computer said, I'm going to have to think a little bit. Okay, how long you're going to have to think? 7.5 million years. 7.5 million years? Yeah, 7.5 million years. Okay, so 7.5 million years later, the whole alien planets came back to the computer to see what is the great answer for this philosophical question. And then when they turned on the computer, they asked, okay, what is the meaning of the life, the universe, and everything? And then the computer says 42. And then the whole planet was looking at each other and said, what? How 42 can be the answer for the meaning of life, the universe, and everything? And then the computer said, look, I really thought about, reanalyze everything, and the answer is 42. But you may not ask the right question, but the right question is going to give you the answer of 42. And then the alien says, okay, what is the right question that gives us the answer of 42? And then the computer says, oh, I'm going to have to think a little bit. And then that's going to take 10 million years, and the planet was destroyed before it gives the ultimate answer, the ultimate question. So that's to show that AI and supercomputers are not going to matter if you don't know how to ask the right questions. So more than ever, we, especially in the global South, will have to care more about asking questions than answering questions. Answering questions, the technical part, the bioinformatics part, is going to be very soon replaced by AI. But asking the question, talking to experts, finding the gaps, coming up with the hypothesis, that's going to be what we're going to left for us scientists. And with that, I would like to acknowledge the team in my lab. So there's a lot of people to acknowledge, including Wafa from Egypt. So I hope very soon we can work together physically. Thank you, guys. Well, we can wait for Wafa to appear because she never appeared in the webcam, but here she appeared. Yeah, we have to update the video for the 2025. Oh, look at her. And that's it, guys. Thank you so much.
Thank you so much, sir, for this insightful presentation.
Thank you, Dr. Helder.
Yes, Rama. I hope next year we can talk about your paper on the microRNA and sex xenomorphism.
Sure, sure.
That's
Okay,
great.
thank you so much, Dr. Hilder, for this presentation. Guys, we will now open the floor for questions from our audience. If you have a question, please use the raise hand feature or type your question in the chat box. Dr. Hilder is going to address as many questions as possible in our allotted time. Okay, thank you.
So there are bioinformaticians there in Egypt, in the audience, that do this kind of stuff using public available data from Egypt.
Ooh.
Only Wafa. Or not?
Can we give your email, Dr. Helder, so if they have any question or they want to do any project, can connect to you? I can email you.
Yes, of course.
Yes, okay. So I will send Dr. Helder email now in the chat box. So if you have any question, you can send to him. This one.
Okay, so we have our first question here. Dr. Jabera, can you please unmute your mic and ask Dr. Hilder?
Yes, Dr. Helda. How are you, my friend?
Bye.
Thank you so much for the wonderful presentation. I'm just thinking curious on, you have mentioned the use of large language model and actual on how to ask components. And we actually implementing a project on artificial intelligence for mental health. And I will do big
For what?
artificial intelligence for mental health.
Oh, mental health. Cool.
Yes, and I'm curious to see on would you, for example, if you want to check on how the large language model like ChatGPT generating the data set and also relevant model set, artificial intelligence model, maybe for prediction of the prevalence of mental health for youth. Is that something that you think artificial intelligence could help, and then our team of experts could just validate what the large language model has given out. Thank you.
Yes, some part of the question was we cannot hear because of internet issues, but I think large language models and AI can be involved in many aspects of mental health from the sociological perspective. For example, one that I'm really concerned is that there is already a AI called Hippocratic AI company that calls you and says, "Oh, hi, Wafa. Here is the nurse. Blah blah blah. I'm talking from the office of Doctor blah, blah, blah. I would like to know if you took the drug that he prescribed." And they say, "Oh no, I thought that was not good, was tasting bad," and then say, "Oh, can you try it with milk?" So that was an AI, and people did not realize that we're actually talking to an AI. And when they test if the AI was more empathetic than the person, they choose the AI. So that means that for therapy, I think AI is gonna be a tool that's already being used, because there are people that's already dates AIs and talk and have friends. But of course, we don't know what's going to be the impacts of having these artificial relationships in the future. There is also a more global humanities issue, which is our ego as a human. I'm going to the data sets that you ask soon, okay? But the ego part is something that also makes me think, because Copernicus showed that we are not in the center of the universe, and that hurt our ego. And then Darwin showed that we did not descend from gods. That also hurt our ego. And now AI is going to show us that we are not relevant or that intelligence like we thought. And how is going to be our mental health when we suddenly lose the meaning or purpose from the work that we do? Like, of course, most people, not most people, but many people use the contribution to work or intellectually as a purpose, and that's gonna impact the mental health. But of course, you were talking about analyzing data sets related to mental health, and there is many ways of how AI and large language models can be used for this. Since the helping you to write scripts to download public available data, and also scripts to analyze this data, process, and even creating dashboards, that's already something that the current models can help you immediately. AI, not large language models, can be used, such as machine learning algorithms, for identify sets of markers that are predicting who's going to die of suicide compared to those that's not going to die, or major depression. And there is a lot of work already on this topic using machine learning models. What else? And I think also computer vision can also be something very interesting to analyze mental health issues, because I think that people with depression or other disorders see the world differently, and I'm very interested to know not only the brain regions that are activated, but also how they physically see the world. And I'm thinking about some stuff with iTrack, but that's another crazy project. And I did not to show you guys, but we do have a lot of known molecular biology projects where we utilize cognitive computing, large language models for medicine, which is not necessarily looking at genes and proteins. I am managing AI from mental health. Oh, you mean like based on talking to youth, like talking to teenager, how can you tell the mental health condition of the person using AI? Definitely this is something doable. Of course, for perform such a for creating such model, you have to have good quality data, and to get good quality data, you need to have good scientists and researchers such yourself to really create the labels, the proper labels that you can use to train the models. But anything that is related to patterns, to analyze and finding patterns, this can be uh, automated or improved using AI.
Thank you so much. That's wonderful.
Thank you. Okay, Miriam.
Yes, sir. Okay, so does anyone else has a question for Dr. Helder? Okay,
Okay,
so
so was really nice talking to you guys. I hope I can physically go to Egypt one day soon, or you guys can come to Brazil.
we would be so honored, sir, to
Yes.
have you here.
Yes. Thank you, Dr. Helder.
Thank you. Bye bye.
Thank you,
Bye.
sir. Bye bye. Thank you all for attending, and please have a nice day.